So good morning, everyone. So we have prepared with Adrian a joint presentation about ILC model systems, cells, and animals. So we would like to focus in 4 aspects today. So we talk about the molecular features about ILC. Already you heard a lot from Matthias, but just to mention some that are important for our modeling. So we will discuss about the challenges to model ILC carcinoma, and then we will go further and discuss about cell lines, organoids, preclinical mouse models, and tumor grafts. So, as already you heard about, and for the rest of this presentation, we will use the term ILC for the models that we will be referring to lobular carcinoma models. And we will use the term NST. This is classically we call it invasive ductal carcinoma, and this is a misconception. So we will use the term NST or IDC in some cases. So don't be confused. But remember that the ILC is the first non-special histological subtype, and NST is whatever has no special form histologically, right? So as you already you heard from the previous speakers, ILC has a peculiar metastatic spread, widespread colonization in sites that normally is not the case for NST tumors, as the GI tract, the reproductive organs, the brain, and even the ocular metastasis has been reported for this subtype. So what is very interesting is that this subtype is exclusively sensitive to estrogen signaling and progesterone signaling, and normally typically express a lot of estrogen and progesterone receptor. So at the cellular level, there is very slow growth of these tumors, of course, as we know, and then what is important is that the adherence junctions are defective. So, and this is mainly due, but not only, but mainly due to weak adherence loss. So, and here you can see the ER staining. This the images from Matthias' publication, 2021, where you can see the estrogen receptor staining, the cells growing in a single file pattern, and they do not express by protein level any EC adherence. So why do we need ILC models? And we will elaborate during the next slides about how we use the clinical information to develop in vitro, ex vivo, and in vivo models. And this is very important in order to be able to replicate the diverse molecular and genetic landscape of ILC, but also to predict efficacy of novel therapeutics, to mimic to study dormancy and metastasis, to mimic the microenvironment, which is very important as we saw also from the previous presentation, and how these cells, they can really adapt in different microenvironments, and to model dormancy and metastasis. So before starting, I just want to say that to paraphrase, to use this quotation actually, that all models are probably wrong, but of course some models are very useful. And in the case of lobular carcinoma, we have unfortunately very few laboratory models for the moment, and this is due to many reasons. Of course, as I mentioned, these are estrogen receptor positive tumors, and we know that estrogen receptor positive tumors are notoriously difficult to grow in vivo. The graftment rate is very low. On the other hand, we have a lack of clinical samples. There is a relatively lower incidence of ILC, and there is under-presentation of patients with ILC in clinical trials. And of course, the third reason is the intrinsic ILC tumor characteristics. that make them difficult to develop as models. So here is a summary of the most commonly used and available models. So we have initially the ILC cell lines, that these are cell lines that derived from material from the pathologies that have been characterized as ILC. However, here we have what we call ILC, classical ILC, or ILC-like cell lines, and we will explain this in the next slide, the differences. We have also, in the lab, we can generate cell lines derived from classical traditional NST models, but we can manipulate the genome and we can have cell lines that now they do not express CDH1, which is the EC-adhering molecule, in the lab. So then we have also the organoids, the ILC organoids derived from patient material. We have also the genetically engineered mouse models. And lastly, we will discuss about the PDX, the patient-derived xenografts, the cell lines that we use to develop also xenografts. And also, we have other animal models that actually they are under development for lobular carcinoma. There is nothing published really about rats or zebrafish. So, to start with, and I don't know if Adrian, you want to continue with the cell lines?
Welcome, everyone. George and I did this together, and so we are going to swap out, and he's going to swap back in for the next part. So I said I would take the cell lines and the organoids, and he would do the animal modeling. And it's great to see everyone here in this great country. So for the cell lines, as George said, there are challenges with the cell lines. There aren't many of them, and they grow very slow, and they're kind of hard to grow in culture. When you look at them, however, they do look a little bit like the human disease. So this is a traditional NST cell line or an invasive ductal cancer cell line where the cells grow and they attach to each other. The ILC cell lines, typically when you look at them, they look like they would in the human in that they are detached from each other because they don't contain EC-adherent, they don't bind each other, and they look like these single cells. But as I said, they grow as single cells, they grow as these discohesive clumps, and they have a very low doubling rate. And I think traditionally, before lobular breast cancer became something of interest to study, people probably wouldn't use these cell lines because they look kind of odd and they don't grow very well. And so traditionally, most research focused on these cells, these NST cell lines. And that's reflected actually in the number of publications for each of those cell lines. So this is a graph of the number of publications on a log scale. So this is PubMed entries on a log 10 scale by cell lines. So cell lines is across here. There's about 60 cell lines here. In blue is the IDC or NST cell lines. This is a challenge when you put two people's talks together, you get different nomenclature. But this is NST here, and then this is ILC. And you can see the number one IDC cell line is MCF7. That has over 30,000 publications now. This is the ILC cell lines. As you see, some of them have no published reports on them and are not publicly available. There are a few that are publicly available, and the best one, which I think is MM134, has about 50 publications. And many of these, actually, most of these publications have come within the last 10 years when lobular breast cancer has become much more appreciated. There just isn't a lot known about these because there just isn't many models to study. And I think that's a huge challenge to the field because you need models to understand the disease to develop new therapies. As an example of this, one of the largest comprehensive analysis of cell lines is something called the CCLE, the Cancer Cell Line Encyclopedia. This was done by Harvard and was an effort to take around 1,000 cell lines and do every molecular profile you can do and make all the data publicly available. And it's an incredible resource for cell lines. When you look at that, this is the Cancer Cell Line Encyclopedia lineages. This is by breast cancer type. You see breast is here, and there are 62 breast cancer cell lines. Obviously, lots of lung cancer cell lines, lots of skin cancer, and breast is number 4. and there are 62 in there. But of those 62 breast cancer cell lines, only 2 of them are ILC. So you have this huge encyclopedia, and again, ILC is basically underrepresented. So you can actually then, so we have this is actually from this review of the Atlas models of breast cancer that came out. That's really the most comprehensive summary of all these models. There are these lobular cell lines here, and shows you what tumor they came from, what their receptor status is, and what their E-cadherin mutation is. But actually, one of the things we really struggle with, and it's interesting speaking after Matthias, because we talk about this a lot, is actually what is the definition of a lobular cell line? So, as Matthias said, ILC is a diagnosis based upon histopathology and growth pattern. That's basically how it's diagnosed. And cell lines don't actually have this. You could look at them and say, okay, they look like it, but they don't have the architecture of the stroma and all those other features that are used to define ILC as a growth in a single line pattern. And so how are we going to actually say they are ILC cell lines or lobular cell lines? And we think about this all the time. This is a huge problem for the cell lines and for the organoids. Perhaps not so much for the PDXs and the GEMs and the mouse models, because you have some form of tissue architecture there. So we could then rely on these molecular features, the CDH1 mutation and other features which are becoming more important in the classification of human ILC. And so this is what George had referred to. So when the RATHER consortium was looking at cell lines, they realized there are a bunch of cell lines here which actually have a mutation in E-cadherin. So they have potentially the genetic marker of lobular. And the RATHER consortium referred to these as ILC-like. They're not labeled as ILC, but they look like ILC. They come, when you look at the tumor they were derived from, many of them are not known. So these were many cell lines that were developed decades ago, and people probably weren't interested in the histomorphology of the tumor or the histology of the tumor at that point. Some of them come from NST tumors. That was the original tumor, but maybe it grew out as a lobular, and some of them have mixed subtypes. So this might actually rapidly, if these were actually ILC, because of their genetic E-cadherin mutation, this would rapidly expand the number of cell lines we have available for research and would presumably enhance our molecular understanding of ILC. And so what we undertook was an approach to basically extend what the RATHER consortium had done and do what the Cancer Cell Line Encyclopedia had done. We took the exact same approach, the same platforms, the same methods, and decided to take all of these cell lines now, go back and get the original clones. Some of them we had to get them from the original investigators, apply every technique, and we have now finished all of this. This is all completed. We, as I said, did this exactly the same as the Cell Line Encyclopedia, so we can now integrate that with all that other data that they have. So now we have ILC versus lots of IDC. We do this in collaboration with the Broad, the group, actually the CCLE group. We do this together with them. So they've taken all of these cell lines now and done drug screening and CRISPR screening. This data all, as it's generated, becomes publicly available, and you'll see this becoming publicly available on their website very soon, because much of this is completed now. And so now you can look for gene dependencies, drug screening dependencies in a large number of ILC versus a large number of IDC, something you couldn't do previously. You can, as George said, you can actually take an NST cell line and delete E-cadherin and make it look like ILC. It's obviously not ILC because it doesn't have the other genetic changes, but it looks like it because it now doesn't have E-cadherin. This is, and many groups have done this. This is an example of our work where we have deleted, using CRISPR, E-cadherin from MCF7 cells and from T47D cells. When you delete it, you can't see this because of the projector, but basically if you look on the screen down at the front, they basically look pretty much like ILC now. They're single cells. They don't touch each other. They have a morphology that looks like ILC. And they have a phenotype that was originally reported by Patrick when he was first studying this. They become anoikis resistant. So their growth in 2 dimensions is essentially unaffected by E-cadherin, here and here. this is E-cadherin positive and loss. But when you grow them in ultra-low attachment, now these cells have an enhanced ability in the absence of E-cadherin to grow, so they have become resistant to anoicus. You can alternatively, you could actually, this title is incorrect, but you could alternatively express E-cadherin in an ILC cell line and revert it back to technically an NST. This was done by Laura, a graduate student who's in the lab. If you want to speak to her, Laura, put up your hand. Put your hand high up. There we go, she's shy. But she has a poster here, and Laura can tell you about this, but basically the E-cadherin, because it's a tumor suppressor gene, is toxic to cells. They don't like it, that's why they got rid of it. So she had to develop a doxycycline-inducible system to do that. So this is doxycycline-inducible overexpression of E-cadherin in the 134 cell line, the SUM44, and the BCK4. You can see it overexpressed here. When you overexpress it, these cells revert to looking like IDC, so they now, upon expression of E-cadherin, get increased levels of P120, alpha-catenin, and beta-catenin. So they start to form the adherence junction again, and they now functionally combine to E-cadherin. That's plated on a plate, and these cells now have the ability to bind to E-cadherin, so they have functional adherence junctions. So two models where you can knock out E-cadherin to look for dependencies, and you can overexpress E-cadherin in an ILC cell line to look for dependencies as well. And then finally, I will just talk about organoids before I pass it back over to George. So growing cells on plastic, like everything I've shown you, is obviously not great because we don't have plastic in our bodies and it's a hard substrate. And there's been an incredible excitement about using these three-dimensional models, as is shown here. This is the first paper from Sachs et al. From the Hans Cleaver's lab, where they developed a large number of breast cancer organoids. A number of them were actually from patients with ILC. And these, when grown in 3 dimensions, these patient-derived organoids, so cultures grown directly from the patients, look a lot like ILC. They have this grape-like structure. They actually have a histomorphology in matrigel that looks quite like ILC. And they have classic mutations. And if you want to read about these are summarized really well in the review again of the Atlas of Models of Breast Cancer. This is a work that we've done on creating. We've created now quite a number of these ILC patient-derived organoids. I think we've done about 25 now. We find many that have these classic mutations, frameshift deletions, stop gain mutations in E-cadherin. They have loss of E-cadherin protein. So this is ILC patient-derived organoids. You see this one has complete loss of E-cadherin and then loss of alpha-catenin and beta-catenin. This one has loss as well. Some of them that grow out still maintain E-cadherin, and we have to better understand why that's happening. But clearly some are classic ILC. And obviously you can't see this, but you would see beautiful E-cadherin staining if you could see it there. And so I'll hand this now back to George for the animal models.
Thank you, Adrian. So I will continue focusing on the GEMs, the PDX and the cell line-derived xenografts. And as you heard many times today, E-cadherin loss is a very attractive target to develop in vivo models. So these are now in complex models, advanced models in a living organism. But unfortunately, E-cadherin loss by itself doesn't really leads to tumor. So although this is only possible if you combine with additional mutations. And here initially, in vivo models that have been generated by Patrick and later from others, they have shown that if you combine CDH1 loss, so E-cadherin, with the P53 loss, or P10, or recently with activating PI3 kinase. So all this information about these additional genes, they are derived from publications from huge high-throughput studies where they have been sequenced hundreds of lobular tissue samples. And here actually you can see the percentage of these mutations found in the lobular tumors in the primary site. So the combination of the clinical information with the in vivo technologies, they have been able to generate conventional, conditional, or somatic GEMS models. These models are retransplantable, so we can put it back in syngeneic mice. They have been developed also 3D and 2D cell lines out of these models and are very valuable for the community. Overall, here I just summarize the pros and the cons of these models. This is advanced models, as I mentioned. They can capture also the extracellular matrix, the stroma interaction with the lobular tumor cells. The mouse model is very useful because it can be easily manipulated. So we can induce overexpression or knockout of whatever target gene is important to study ILC. They can metastasize all those in clinical relevant organs, although most of the time they metastasize in lungs, or we are lacking, and now I'm going to the cons, we are lacking some metastatic sites that we often see in the clinics, like leptomeninges or ovarian metastases. And many times they have fast growth, which is not characteristic of the lobular subtype in the primary site. So, but again, these are very useful models in vivo. And the next part is, again, in vivo, is to develop xenografts or PDX out of cell lines or freshly dissociated tumor pieces and put in the mice. So we are taking this tumor material either from primary breast or from the secondary ILC, derived from different metastatic sites like pleural effusion, leptomeninges, ascites, ovarian mets, and others. And many of the cell lines, actually, that you saw before on the table have been generated out of pleural effusion. This is a very classical characteristic of a cell line, of the existing cell lines. So these cell lines we can cultivate, hopefully, making 2D in vitro models, and this is how they have been developed as the few cell lines that we have in our hands for lobular carcinoma as a 2D. Recently, as Adrian mentioned, we have the 3D organoids that are currently a very active field of research. And if we want to go to in more advanced setting, we inject these cells in the mice. We inject in the different injection sites. Could be either INtraDuctal, could be in the fat pad, sub-Q, or other anatomic sites. And this is the way we develop, they can develop metastasis spontaneous, what we call spontaneous or experimental, and I will explain later in the presentation. So here, I just want to mention some pros and cons of these models for the cell lines and the PDX. First of all, it's very easy to work with the cell lines because are available. You can take it from a lab, you can buy it. It's very well characterized, and you can study endocrine responses, for example, in vivo. The PDX, however, these cell lines are not heterogeneous, so the PDX are more heterogeneous, and of course they can maintain the clonal diversity, and they can be used for therapeutic, testing therapeutic drugs. So the big issue with these models, it's are very time-consuming concerning to the in vitro and ex vivo. So these experiments, they can take up to a year, especially if you want to really study mechanisms of metastasis. So it needs really a large amount of resources, except from time, and most of the time we put these cells or fresh tissues in immunocompromised or immunodeficient mice. So here is an example, it's a table of, there are very few cell lines, and there are very few cell lines also available that are ER positive. So here is an example of a recent efforts that we did in the lab to generate xenografts out of the two available, actually, well-characterized ER-positive cell lines. And also in the past, there is the triple-negative cell line that Matthias has been developed that has been reported in the literature that has been grown subcutaneous. So these are cell line-derived xenografts, and we have the PDX that here is a summary of the publicly available PDX that you can find if you look online in different data sets that are partially characterized. Some of them, they have been characterized further. However, we are really lacking complete information about the ECAD-hearing status, as before Adrian mentioned about the very comprehensive analysis of ECAD-hearing status for cell lines. So, this is unpublished data from Andreas and Steffi lab that actually they took some of the cell lines, then they put it in the fat pad, they inject them back in the mouse in vivo in the presence of estradiol pellet. That helps the growth of the cell lines. And this helped to, so here you see the time scale. So what I mentioned before, it really takes up to a year to develop tumors and metastases out of these models. And here you can see they are able to detect metastases at different sites in brain. And this is the histology of these metastases. And some of these tumors retain the estrogen receptor positivity as they metastasize, which is a very interesting feature. So we in the lab also recently, we have developed the approach of INtraDuctal injection. So we take directly in the cells and we inject it inside the duct in a completely different microenvironment comparing to the other methods of injection. And in this way, we are able to have high engraftment rate for ER-positive tumors in general, and ILC was also not an exception. So we are able to retain estrogen receptor positivity. And with these methods, actually, you can do the experiment without the need of supplying estradiol supplementation. So you can also perform easily endocrine studies, for example. And so I'll show you one example. I just want to highlight here again the difference between NST and lobular carcinoma cell lines. So here what we see the, again, the morphology of those. These are milk ducts. So this is mouse milk ducts. So we see how morphological, if we inject NST cells, these cells, they will grow, they will dilate the milk ducts, but they will sparing the tip of the ducts here. So they don't really fill this area. However, in contrary, both lobular cell lines, as you can see here, they have a completely different pattern. They form a human-specific structures that could resemble the tactile terminal lobular units, this grape-like growth. That it's another characteristic that points to distinct morphogenetic properties of lobular carcinoma. This you can see further when you inject also primary tumors, lobular tumors that are more heterogeneous. You can see, and you can see better if you want on my computer, this image you can see, you can get really a characteristic of lobular carcinoma as a single signet ring appearance. And so here we see two different histomorphologies. And the estrogen receptor expression, importantly, is retained in these patient-derived xenografts and with different distributions between the two different morphological cellular subtypes. Importantly, these models we have shown that are responsive to estradiol and progesterone. So for example, the 134 cell line can grow, the estradiol progesterone promotes the growth significantly. And here you can see also the staining of progesterone receptor, how it's affected by the estradiol. This is very important because we are lacking this model that can be estrogen responsive. And finally, for this part of the model, we have proved that this model is also metastatic. So actually the metastasis is a late event. It's 9 to 12 months growing these tumors in the mice after transplantation. And they are going in a very clinical relevant spots like the ovaries, the adrenal gland, the brain and other organs. So we can study metastasis this way. So to finalize, we have the advanced metastatic models that can give spontaneous metastasis. So what I described is all spontaneous metastasis that can also be the result of the genetically engineered mouse model or the PDX, so that you can have tumors in different metastatic tissues. We also have actually for lobular carcinoma we don't have, but there are, for other breast tumors, we can have experimental metastases that we can inject directly tumoral cells intravenously or intracardial in order to get metastasis faster. Because as I mentioned, this takes at least 12 months to be able to study metastasis. And this is something that is missing for lobular carcinoma studies. But of course, we have always to remember that these are all models, and we also have the human as a model, which is the most relevant. And there are recent efforts to have the programs that they call rapid biopsies, that you can actually study the tumors, the actual tumors, and comparing with the metastasis and get information about transcriptomic, genomic, proteomics in the most relevant content. And there are two efforts. There is from Christine Desmond lab and Adrian and Steffi lab that are currently running. Of course, from these tumors, we can also, again, take material and put in the mouse and do the PDX studies. And here is an example from Adrian study. I don't know if you want to mention something on that.
Yeah, I can quickly say, I think this is really important. We need to study the tissue, and it's very hard, as everyone knows, to get tissue to do our modeling research. And so this is an example of 24 autopsies that we've just recently done. You can see the subtypes there. Unfortunately, only two of them ILC. One's an ILC and one is a mixed ILC IDC. We need to have better representation of that to try and understand the disease. But we do, I think this is going to be invaluable for understanding the biology of the disease and then developing even better models to try and develop new therapeutics.
Thank you. And just to give you the last slide before the summary, just to give you an idea that to develop models takes time. So the two cell lines that I was mentioning, the most widely characterized for lobular carcinoma are coming from 70s, as you can see here in the time scale. So it's really very laborious and takes time to have the first, for example, xenografts. To develop effective xenografts, it took more than 40 years. And here are the examples of the first gem or the first triple negative cell line. And you can appreciate this time scale. So to summarize, we have mouse models that we can study the ILC, including the microenvironment. There is no single model that can recapitulate the complexity of the disease. We are still missing, and we really need more models that recapitulate estrogen progesterone receptor, ILC, positive ILC, and immunocompetent models that we can combine with engraftment of ILC tumors. We need, and there are many efforts for organoids to develop further more organoids and PDX to test new therapeutics. We need definitely to increase the quality and the quantity specifically of the lobular models. And importantly, to do that, we really need to collaborate, and here I just want to point the importance of collaborative effort in this field. So with that, we finish the presentation, and I would like to acknowledge all the people that have been contributed to this study for lobular carcinomas. So my gratitude to Professor Catherine Briskin Lab that I'm working on for the enthusiastic support for these studies. I mean, we have the wonderful collaborators within the ELBCC network, which is very important for developing further lobular models. And of course, we like to thank also the patient advocacy and the humans that really donated their invasive lobular tissue for this study, and their families. And the next slide.
Yeah, and similar to what George said, you know, this takes a village to do this, obviously. I want to thank the lab. Some of them are here, and you can interact with them. They have posters here and are giving talks here. And obviously the advocacy groups and the patients and everyone who's participated in all of this research, which I think is really important.
Yeah, and thank you all for your attention.